Hello friends, welcome to lecture 1 of week 11. So in this lecture, in the 3 lectures that we will be doing in this week, we will be looking at the bacterial genome sequencing, assembly, annotation, and also the detailed comparative and metabolic analysis. So in this first part of this lecture, we will look at what are like bacterial genomes or prokaryotic genomes, and what is their genome size, what are the best methods to complete their genome sequencing or prepare their draft sequences, and the difference between complete and draft, and also how we can taxonomically annotate these genomes and how to identify these genomes. So these are the things that we will cover in this lecture. So basically our planet is the planet of microbes, and actually all the life form that we see today is originated because of the activities of the microbes that change the Earth's atmosphere. And even today, like, you can find bacteria in almost every corner of this planet, and their number is extremely high, and like billions, and basically we know only 0.5% of them, right? So also, like, it is limited by the culturing approaches, because we need to understand that in what condition they will grow. They might be dependent upon other microbes for their survival or getting something from them, or the typical physical, chemical, biological condition that they need for their growth, right? So that is why it is limiting to grow all the microbes. But this field is evolving very fast, culturomics and followed by the genome sequencing, and in addition to metagenomics, which we have covered already. So basically these microbes that we want to sequence, or we are discussing today, is particularly bacteria, but the method that we will see can be applied to smaller other micro eukaryotes also, such as fungi. Okay, so basically we want to achieve from any bacteria that we see is to get their complete genome, that is the list of genes and the entire set of nucleotides and their respective order, and from which we will predict the genes and get the list of genes, and further analysis will be done by that. So basically microbes are something which are microscopic, which cannot be seen by naked eye and need a microscope to see, and among them there could be micro eukaryotes also, which we cannot see, and they could be single cell, the typical bacteria, or it could be even multicellular sometimes, like they could be like cluttered together or clustered together, like E. coli cluster that we see today here. So basically we are dealing with the single cell, or basically the colonies of these bacteria, and we want to culture these. The databases such as NCBI and the other reference databases that contain Genome Online, for example, database, so they are now getting full with getting submissions from all over the world. So many microbes, prokaryotic genomes are getting sequenced, and you can see that almost in, like, if you see 2021 paper, it had almost 2 lakh bacterial genomes and archaeal genomes, and almost 150 million proteins from all these genomes. And you can imagine that keeping these genomes and doing the annotation and the analysis is really a time consuming and a challenging job. Like as of today, if you see the current status, so basically there are almost 4,18,000 bacterial genomes which are available in the Genome Online database, right? And about 5,000 archaeal genomes. So basically this number is increasing so fast. Like we saw 2 years back, that RefSeq paper that we just saw, it was 2 lakh about, and now it is like 4,18,000. So it is almost doubled in 2 years. So what is a genome sequencing project or bacterial genome sequencing project, and what we should do, and why to sequence something? So basically, first of all, you should have a bacteria to sequence, and the source of bacteria could be a part of your research, that pathogenesis or use in industry or some molecular biology application. So that could be one of the bacteria that you have isolated, or it could be of your interest, or you have isolated from a particular environment, for example a hot spring, or near a volcano, or basically some ocean or deep sea. So there has to be something unique about the bacteria. right? Or it could be like something from the gut, human gut, or basically skin or oral cavity. So that maybe becomes your starting point how to choose a bacterial genome. Okay, and then first of all, you have to do the confirmation of the species by looking at some molecular markers such as 16S ribosomal RNA. So you will extract the total DNA, amplify that particular gene, and then go for the sequencing, so that you know that what you are sequencing has not been done earlier, or even if it is like a kind of partial sequencing, that even at the species level you can find a bacteria, but your 16 ribosomal RNA is different a little, okay, and other species was found from soil and your species is found from, say, some organismal environment such as human gut. So you may like to go for it, okay. Then again, like from which tissue or part you have extracted, if it is host associated that bacteria, right. Then you can go for like either conventional way of extracting the DNA or using a kit, okay. So then you have to assess the quality of the DNA and RNA if you are also doing the transcriptomics, which is also equally important. And then decide the sequencing strategy based on the genome size estimation. So if you've identified the bacteria, you will know that what should be the approximate genome size of that bacteria, and you may go for about 200x or more with the like traditional or basically traditional in case of bacteria, the Illumina short read technology, or you can go for like 30 to 50x with the long read technology. So other things is the whether you want to do the assembly and annotation on your system, or you want to use a pipeline which is already available. Then you would like to go for the phylogenetic analysis, where you would like to compare it with the other bacteria and see that in that phylogenetic tree where your genome is lying, right. You would like to construct the metabolic pathway of your bacteria, so that you know that what all metabolic functions it can do, for example, what all amino acids it makes, for what it cannot make, and it may be importing it from outside, right. So these things you will find by metabolic pathway construction. Then overall, like whatever results you get in terms of genes or any new finding, you would like to further interpret it, do some more test or biochemical analysis, or do some supporting analysis computational, and then report the new findings as a manuscript and submit the data as NCBI, because you need to get an exception number for your genome, so that you can submit that genome, and so that like others can also access it later. You can allow the permission today, or you can set a date on which the data will be released, and that number, accession number, is also important for the manuscript writing purpose. So the challenge today is that there are so many genomes to sequence, and we are limited by our technology. If you use like Sanger reads, it will take forever to complete one bacterial genome also. If you use short read, the Illumina technology, it is a read short, and you may end up having a good draft genome but not the complete genome. Even if you use the long reads, only the long reads, nanopore or PacBio, possibility is that you may end up with some gaps, and you still want to little more sequencing, or do primer walking or other methods to complete that genome, or improve the quality of that genome using the Illumina sequencing. So just to get an estimate of the bacterial genome, so basically the smallest bacterial genome that is found is about 1 million base pair, right. That is a free living bacteria. There could be like endosymbiotic bacteria, or very small size like 0.5 MB also, but those are mostly in parasitic or symbiotic mode, right. But the smallest free living is about 1 million base pair, and the largest and basically you can see that this bacteria had only 182 protein coding genes, and okay, so basically you can see this symbiotic bacterium, which is the smallest one, is only 159 kb and only 182 protein coding genes, and it is much smaller than what we can imagine that for life, like to sustain only 182 genes like were found sufficient if you think of the idea of a minimal genome. But still you can imagine that it might be using a lot of host proteins, amino acids and other machinery for its survival, right. And then typically you will find the bacterial genome to about 4 million base pairs, right. Okay, and the largest bacteria that is ever reported so far is about 14 MB or 14 million base pairs containing, which is like almost 1.75 MB more bigger than the previously known bacteria, that was S. cellulosum. Okay, so basically people are finding new large bacteria also, and but the typical range would remain from about 1 million base pairs to about 14 or 15, and the average is like 4 to 5 MB. Then when you are collecting a bacteria or isolating a bacteria from a location, you have to know the source of the bacterium, like host, or in terms of physical location like the coordinates from where you have got like your sample, other physical conditions in that location such as pH, TDS, total TDS, or oxygen concentration, or temperature. These things you should note. Then when you have culturing, then you have to define the media in which you cultured it, and what were the colony characteristics, and then you identified the strain using the or the species using a 16S ribosomal RNA approach by Sanger sequencing. Okay, what DNA extraction method or purification steps you have performed, and then basically going for the genome sequencing and submission at NCBI. So all these things, these you have to provide when you are submitting a genome at the NCBI. So typically your whole genome sequencing of bacteria process would look like this. So you will be having a bacterial culture in hand in the form of a Petri plate or in a culture tube, right, liquid culture. Then you will extract the DNA, right. You will prefer to extract the high molecular weight DNA if you want to go for the long reads, right. And even like high molecular weight is good for even long read or short read. Okay, then basically you will be fragmenting your DNA for like short read sequencing or like the defined reads which according to the protocol. Okay, then if you have little lesser DNA, you can go for PCR. Otherwise you should do a PCR free approach and basically go for the direct library preparation using either Illumina or Nanopore or PacBio library preparation kits, right, followed by the sequencing and analysis. And then when you have completed the assembly, you will be ending up with these kind of terms such as the read, which is basically the sequence that comes out of the sequencer, like depending upon the sequencer. For example, the Illumina sequencer may give you a read length of 150 base pairs. Nanopore can give you something like 2 kb to 10 kb to even longer than that. PacBio also similar lengths. Sanger can give you 800 base pairs. So like this is the length of sequence that you get originate from the sequencer. Then the assembly that you want to connect the reads based on overlaps and using De Bruijn graphs, or that we have discussed so many times earlier, or those kind of approaches, and then assemble it into contigs such that you have exhausted that there is no overlapping chimer or the fragment that can fit into that contig. Okay, then scaffolds, when you have a lot of contigs, you want to arrange those contigs in the right order and orientation, so that you can connect the long distance contigs also into a scaffold, which considerably increase the length of the draft genome or N50 of the draft genome. Then some units like kilo base pair, kb, 1000 bases, or million base pair, mb, 10 lakh base pair. So basically these things are these words you will see when you are doing the sequencing, right. So typically you will start with collecting the samples from an environment, or if you already have the sample like this in a plate. So if you are starting from here, you have to isolate the bacteria in a Petri plate. You have to get the pure culture of it, like this could be one pure colony. Even you would like to take this colony and again go for the streaking on a new plate, so that you get very distinct and well separated colony, because very close colonies may lead to contamination or mixing up of bacteria, and there could be some bacteria which are growing together, mixed culture. So you should get as pure colony as possible to pick up for the DNA extraction. Then you will use a kit or the protocol defined user extraction, or basically a conventional user DNA extraction method, followed by the library preparation. And while you are doing, after you have done the DNA extraction, you will take some DNA and do the amplification by 16S ribosome RNA gene and go for the identification of the species. And the library that you have prepared, you will go for the sequencing. So basically, the sequencer that you are choosing, either it is Illumina or Nanopore, they produce like for example a Illumina NextSeq 500 may produce almost 40 GB of data, like with the smaller flow cell, okay, mid output. So basically in 40 GB you can mix almost 40 genomes of bacteria if you go for 1 GB per bacteria DNA outcome. So you have to multiplex or mix multiple bacteria together, so to get advantage of the total sequencing capacity. So you will be using barcoding and multiplexing, which you will be later demultiplexing, okay, and followed by the DNA analysis. And it is important to basically get the pure culture of the bacteria before you go for sequencing, because in the mixed culture there is a possibility that it could be too, like if it is not very well separated colony, it could be possible that multiple bacteria that grow depends upon each other, or basically the bacteria were not well separated, or it was motile bacteria also, that could be one problem, okay. So try to get as pure culture as possible, and that pure culture that you have used for the genome sequencing of the bacteria is also needed when you go for the culture collection and submit that bacterial culture to a repository such as something in IMTech or MTCC and like that, right. And then you also have to report the morphological, biochemical and genome characteristics of that genome, how the colony looked and what was the morphology of the colony and these things. And basically all microbes are different, and you have to define their strategies accordingly based on your experience or the environment you isolated it, or like what you learn from that microbe. And it could be like some are very abundant, easily like you can get in the colony or in the plate, but from any source, like if you go for a lake also for the extraction of or isolation of bacteria, the ones which are more abundant and easily culturable will grow very fast, and those which are rare but still important for that environment, okay, you may have to spend a lot of effort and pick that bacteria. Okay, so basically for pure culture you can do either pour plate or spread plate, and basically when you are isolating from an environment, okay, both methods can be used, but like spread plate is better because in that you can like spread it well, and also you get the chances of getting the colonies which are more aerobic is higher, whereas in case of pour plate you can, since you are mixing the culture with the media which is like lukewarm when you are mixing, so chances are that some will go beneath the media also, and like those which are like facultative aerobic or which requires less oxygen may grow there. So you have to look at the various methods, spread plate or streaking for isolating the bacteria, right. So now the colonies that you get for these bacteria, again, like could be very different. It could be very small pinpoint or very large or rough or smooth, or having viscous, or having some kind of capsular formation or sheath, right. So for example, or you can see that this is very small pinpoint colonies, or it is very circular, irregular, okay, mycelia, so curved, convex, right. So different type of colony morphology could be there, which you have to take pictures and report when you are submitting that genome and also writing the paper for that genome. Okay, then basically this part we have covered mostly, that so many times that when you have the bacterial DNA, you have to fragment it by shotgun sequencing, and then you are sequencing that using the sequencer which is producing read, okay. So it could be single end read or paired end read, like you are reading only from one end, so it will be single end read. If you are reading from both the ends, it will be paired end reads, and then you will be mapping that against a reference genome, or you will be doing a de novo assembly, in the case of in your genome. Okay. So, basically all the three generation of sequencing, first, second, third, are useful for genome sequencing, because the first generation sequencing, that is the Sanger method, will be producing like short reads, and it could be easily accessible to you, and the running cost is also not high. So, basically it is very important for the species identification. So, once you have amplified the 16th ribosomal RNA gene, you can use a Sanger sequencer which can produce read lengths of about 800 base pair, right. So, in that way you can easily cover, like for example you can see so you have to, like this was a 16S ribosomal RNA gene, right. So, you can read it from both sides, okay, with an overlap, and basically both sides will be so this gene is about 1.5 kb, okay. So, using Sanger forward reverse read, you can cover 800 base pair from both sides, and this way you can cover the entire 1.5 kb gene, right. So, basically this Sanger sequencing is very important for the species identification and moving forward to the genome sequencing. Now, you are moving to the second generation sequencing, which is Illumina or Ion Torrent, which will be producing the short reads for the genome, which you will be assembling using the other like ABIS or SPADES or these kind of assemblers, right. So, you can choose any of the instrument, like Roche is not no more existing, but Illumina and Ion Torrent are available, and Illumina is the gold standard with lesser error rate. So, generally like most of the data that you see today is from Illumina short reads. So, you will be using this for producing the short reads for your genome. Okay, so this is the Illumina sequencing technology that we have discussed earlier in detail, and basically the idea is that it will be, you will be constructing a library, and you will be using the any of the Illumina sequencing platform. There are so many available, right, and you by the bridge amplification and cluster generation, and followed by the sequencing by synthesis, you will be getting the sequence reads, okay. So, like I said, there are so many types of Illumina sequencers. You can see iSeq, which can produce about 1.2 GB of data, which is very good for a single genome. You can run single genome very easily, right, about 400 million reads per run. MiniSeq, which produces about 1.65 to 7.5 GB data. You can mix almost 7-8 genomes here, right. Okay. NextSeq, which can produce like something from 16.25 to 120 GB of data, which will be very good for sequencing a lot of genomes together, right. Then MySeq, about 15 GB data. HiSeq and NovoSeq will be like too much for a bacterial genome, but definitely it will be running by some service provider, or if you have it in your lab, then basically you will be mixing or basically multiplexing your samples, genomic samples, including the bacterial or multiple bacterial, along with the other sample that you have for sequencing. So, because it will be producing like almost 6,000 GB of data, and you can easily cover almost 6,000 bacterial genomes in one run in NovoSeq. Okay, so basically if you just see like iSeq, which produces about 1.2 GB, just good for a genome. So, it will take about 1 to 2 hours for DNA extraction and target enrichment, and about 2.5 hours for the next era DNA flex library preparation, which is also fast, right. About 17 hours for the sequencing, right, and about 1 hour when you have uploaded the data on the base space, which is the basically kind of Amazon cloud service by the company. So, it will be doing the standard genomic analysis for the data and reporting you the results in about an hour. So, basically today we have the pipelines ready for quicker, faster, reliable, and robust sequencing, as well as the assembly and also the analysis of the genomes. Okay, like in iSeq, the runtime is about 19 hours, whereas MySeq may take about 55 hours also. Both of the instrument like iSeq and MySeq are very good for bacterial genomes, okay. iSeq produces 1.2 GB, MySeq 15 GB. Okay, so basically in this you can mix 15 genomes. This is good for one genome, and like that you can decide based on the availability also which sequencer to go for. Okay, NextSeq 500 we have already discussed, basically two options, 40 GB or 120 GB, and there are so many library preparation methods. So if you have dealing with lesser amount of DNA, you can go for NextStar XT library or TrueSeq Nano library, which you can start with 100 to 200 nanogram of DNA. So depending upon the genome or DNA in hand, you can go for the NextSeq 500 sequencer also. Then for the long reads, you can use a USB based MinION device, or even if you have so many samples, you can go for Gridiron or Promethean also, right. So basically per MinION device like this kind, you can get up to 20-25 GB of data, which is again good for about 20 to 25 genomes and bacterial genomes. And the technology here is not polymerase dependent, but basically DNA passes through the nanopore and changes the flow of the ions through that pore, which produces a signal which you detect and call the basis. So this is we have covered earlier also in the case of eukaryotic genome, this technology. And basically the problem with this would be it may have a lot of errors in the like 10% error. So you have to do a like higher amount of sequencing to correct those errors, or you have to support it using the Illumina data, short read data, by mapping it to the long reads to correct the errors. Okay, so typical like bacterial genome sequencing assembly pipeline for the nanopore is like shown here, that basically long reads assure that the genome that you are doing will be nearly complete or complete with very little gaps. Okay, and it also does not need like too much dependence on the assembly methods, because assembly method itself sometimes create a lot of errors, right. So basically this is using the OLC based methods, and that we have also discussed in case of eukaryotic genomes, and maybe more reliable, right. So first of all, the DNA will be extracted, and the company suggests a lot of things such as the QIAGEN genomic tip for high molecular weight DNA, then basically the purification by agent coat MP or beads, and you should also use a short fragment eliminator kit, so that shorter fragments are gone and whatever you are left with for the sequencing are the longer fragments, so that your N50 or basically the average read length that you get from the sequencer are in the higher side, right. Like this you are getting like mostly reads in this range, right, 10 to 25 kb and almost reaching almost 100 kb in length. Okay, then basically the higher molecular weight DNA is preferred in case of long read sequencing. And once you have prepared the library by using the adapters and ligation, you will load the library, and it will take about 24 to 48 hours to if you are going non-stop and using the flow cell completely. So okay, so basically nanopore is a very good way of and basically since you have about 20 GB data that you can produce, you can put almost 20 or even more samples together by the barcoding and multiplexing, and later on you can resolve these. Okay, so nanopore gives you the long reads which you can then assemble using the Fly software, assembly software, which we have already discussed earlier. It deals with the FASTQ files, and the method is almost same as what you do in the prokaryotes. And then you can do one round of genome polishing by Medeka software, or even you can use the short read data if you have for the polishing steps, and then complete the genome followed by the genomic analysis. Not only this, basically nanopore can also allow you to recover genomes from metagenomes, because once you are producing long reads, the chances that the read is coming from a single cell or single bacteria is much higher, right. And that is why like long reads can be used to complete the genome assembly if you have sufficient overlap, and you can basically bin those reads into various genomic bins and construct the draft bins and Then after the polishing and further steps, you can go for the final bins, which could be representative genomes or kind of mags or basically a high quality draft genomes. Like nanopore, PacBio can also be used for the long read sequencing, or basically producing long read genomic reads from the bacterial genome sequencing projects, right. So this process uses a polymerase and a ZMW. So basically your fragment hairpins are attached to both sides, and then it is opened and a polymerase moves in this way, so that it completes that single strand molecule sequencing, right. So this also produces, like PacBio Hi-Fi kit produces almost 99.9% correct data, and which is very good for a long read data and can be used for a good quality bacterial genome sequencing. Okay, so basically there are two parts for a genomic project. One is using the 16S ribosomal RNA, where you will be amplifying and doing the Sanger sequencing, and knowing that what bacteria are dealing with, and what is the like genome size and those kind of estimations, and followed by the whole genome sequencing that you can do by short read or long read. Okay, so basically just to give you an overview again of the steps. So the first step will be the extraction of DNA from a pure culture, right. Followed by either a single type of sequencing like long read or short read, or a hybrid sequencing where you will use both the long read and short read like ONT and the Illumina reads together. Okay, then you will be assembling the long and short reads separately or together, okay, and constructing a hybrid assembly. Then you will be polishing the long read assembly using the Illumina reads, because long reads are prone to errors, and so long read and short read polishing, and after that you will be doing some manual curations to improve the quality of the genome as needed, and also to like define the origin of replication and rearrange the genome coordinates according to that origin of replication. Okay, so basically now after you have done the sequencing, you will be dealing with these kind of terms, that is completely sequenced. That means the complete genome or identity of all the bases is known and there is no gaps in the genome. In the draft version, it could be high quality draft or low quality draft depending upon the number of gaps, and it is in like different contigs or scaffolds, right, and with a lot of gaps in between. The coverage, like if I said like 200x coverage needed for short read. So for that there is a typical formula which is n into L into G, that is the number of reads into the length of the read divided by the genome size. So this can help you to get the number of what should be how many reads you should generate of a particular length to get the X coverage for that genome. Okay, so basically you will be dealing with short reads which you will be assembling together and making a contig like this. You can see that basically there could be several short reads based on the overlap of those reads using KMR based approach, that is a de Bruijn graph, right, velvet or ABACE, okay, or spade. So basically this will be helping you to assemble contigs out of it. And then there are again like two things, like either you are going for a de novo assembly, that means you do not know the genome or there is no other genome which is known or for that species, and you are assembling the reads, constructing the contig scaffolds and trying to reconstruct the genome from scratch. Or you already have a template, for example E. coli infections are spreading, so you are isolating new strain and comparing with the known E. coli strain and looking at the differences. So in that case you will be mapping the new genomic reads into the previously existing E. coli strain and comparing, and basically after that mapping reconstructing the genome. Okay, and in general like hybrid sequencing and assembly approach is the best, because you are using the features of both long read and short read for getting a high quality and highly contiguous draft genome. So basically hybrid genome sequencing assembly we have already covered in case of eukaryotic genomes and today also briefly. So I will not go into details of this, but basically combination of these two will be the best way to go forward. Okay. So now what is a draft genome or a complete genome? So basically you have started from something like this, which is a circular bacterial chromosome or a plasmid, right? Basically you have fragmented it, right? And again, if you are able to reconstruct the same genome, right, so that is the complete genome. However, in most times you may not be able to do this, and you will be having like very long contigs, right, or scaffolds, like for example this way, like this genome is now in 5 pieces, right? So we do not know like what is there. So basically typically gaps, we do not know like can we really number these are 1 2 3 4 5, or they will follow some other order, or this will be 1 and this will be 2 and like 3 and 4 and 5 and like that. So we not know, also do not know the order of these contigs, right? So this is a draft genome. Typically like less than 100 pieces is a good draft genome, but people have, like if you see in literature, there could be like more than 100 pieces also, and so many types of draft genomes are available, right? So basically at this point you should see that how you can do a better assembly or reach to a more contiguous assembly. Okay, so one of the tools to do the bacterial genome assembly is SPADES, which is St. Petersburg Genome Assembler, and we have also used it in the case of MetaSPADES, which is for the metagenomic data. So basically you can download this from this site and also from GitHub, that I'll show in the next slide. And the latest paper that describes the SPADES software is available here, which will tell you like the algorithm, the advancement of SPADES in comparison to the other genome and assembly softwares, right? And also like how to use it. So from GitHub you can download SPADES, okay, and the same way like you installed, we have now in the 11th week, so we have done the installation and also examined like how the different softwares are installed. This will also be like similar way you can do, okay, and once it is installed you can use it, right? So to use it, basically you can say metaspades py help, okay, that will display all the commands. Otherwise you can say like spades, which is for the bacterial genome, this is for the metagenomes, right? So -1 is the forward file, you have done the paired end sequencing in this case. So forward file, reverse file, okay, R1 R2, okay, total number of threads, k-mer if you want to define something, or you can do iterations and select different number of k-mers, okay. What should be the thread offset? Generally 33 is preferred, and because you want to use high quality reads for this, and the output folder name, right? And then later on, once you have done the assembling, you can check the assembly statistics by using the quast tool script, where you will be giving the folder which you have generated here, right, which contains a file called context.fasta, okay, and then the k-mer size that you have defined earlier, and number of threads. So basically it will be generating a report in this folder, right, that for the k-mer size 21, it will create a folder called quast k1 k21, and then you can say cat this report.tsv, which will show you the statistics or the N50 number of contigs, right, GC percentage, etc. of the bacterial genome that you have assembled. Okay, so compared to the tools which are available, so it can SPADES has very good in terms of time taken, the RAM which is used, and basically in case of either an isolate or a single cell. So this was tried on a 16 thread server, which is 16 core system, which you may be easily accessing, right? Intel 2.27 gigahertz processor, which is even these days people are dealing with 3 or 4 gigahertz processors, okay, 8 GB of RAM, which is also minimal. So that way you can complete the complete analysis in like about 20 minutes for E. coli, and okay, so basically this does not have a lot of computational requirements, and you can easily run the bacterial assembly of on your local system, right. And if you see, like in the past, the reference genome assemblies by using all these sequence technologies and the assembly pipelines, it is increasing so fast that you can see that in RefSeq, basically per year, like 2009, if it was only one assembly added, so in 2020, like 24,000 new assemblies were added, and total assemblies reached to almost 2 lakhs. Okay, so which means that there is a very rapid rate of genomes which are completed and submitted at RefSeq. Okay, so basically once you have done the genome assembly and sequencing and assembly, you will be reaching something like this. But if you note here, there is numbering which is 1 to say 1.1 million, right. So how to say that which one is the first base pair in your genome, because the bacterial genome is basically circular, right. So it is like a wheel, okay, like just an example that you do not know like which one is the first spoke of this wheel, right, or first you can say the stick of this wheel. So because the wheel you can rotate, and so basically the first position is assigned to the OREC gene, or the gene from which the which has the origin of replication. So that location is assigned the first base pair, and generally that gene is 1 to 1317 base pair, like okay, and all the genes that you found, or basically the genome numbering is according to that gene, okay. And there will be one side which will be exactly opposite to the origin of replication, that is the termination site, because bacteria generally follows bidirectional replication, so there has to be a termination site also. And then you will be liking to generate a kind of the circular image for your bacterial genome, where you will be finding maybe the GC content or GC skew, arranging the genes in the forward strand and the reverse strand in basically concentric manners, and the pseudo genes or tRNA, ribosomal RNA that you found from the genome, right. Because genome is circular, so you in circle you can say, you can show the genes, basically you can see different colors for these genes also, like blue, purple, pink, etc., based on the type of function they are involved in, which also tells another thing, like when you are connecting those genes in the circular fashion, that which region of the chromosome or the bacterial genome is more rich in metabolic genes or signaling genes, or any other kind of genes, right. So constructing a genome map after the genome assembly is one of the outcome of your genome analysis, okay. So the first gene that we consider is the origin of replication gene, that is the OREC gene, and we would like to identify that gene and renumber all the genes according to that gene, right. And GC skewing is basically one thing that you observe in these genomes, that it is not that the percentage of G and C will be same on both the strands, right. Okay, so basically it is calculated as G minus C divided by G plus C, and it is more so the strand which is having more number of coding genes, right, it could be the forward or reverse in the case, will be having more of the GC skew because the coding genes prefer like more G's as compared to the C's. Okay, so then the another part of your after you have done the genome analysis is the taxonomic assignment. You want to identify that what bacteria it is, and right. So it could be done at the first thing also, or like just before the function annotation step, where you want to identify the complete or the correct name of that bacteria. You can also, if it is a very novel bacteria, you can also propose your own name for that, even the genus species name, but it is not likely that name will be retained, and later on NCBI and other agencies which deals in the nomenclature of the bacteria, they may assign it to some particular group based on the family or order to which that genome belongs. Okay, so basically you want to assign a typical genus and species name to the your genome, right. And then basically you can know that where your genome is lying in terms of, okay, this is this species belonging to this genus and this family and basically this kingdom, which can tell you a lot about the relatives of that bacteria, distant or far, right, their characteristics, and also the features that are inherent in that particular domain or kingdom. So typically 16S ribosomal RNA gene, either complete or parts of it, like this could be any variable region from that gene, is generally used to identify that bacteria, because 16S ribosomal RNA gene is present in all the bacteria, so it is conserved across the bacteria, but still it is variable. That means there are regions like this variable region 6, 7, 8, 9 from V1 to V9, okay, which are of different sizes that vary a lot between even the bacterial species, and much more when you go like further or higher in the taxonomic level. And the constant regions help you to amplify these kind of variable regions, because you can easily because they do not change much, okay, and you can make primers from these regions and amplify this particular region of variable, okay, from forward and reverse primer can be made from this region and this region, which can amplify this particular region easily, right. So that is why 16S ribosomal RNA is very important for the taxonomic identification and characterization, and also later on based on this, and also the other genes which are conserved genes, such as the other ribosomal genes or the ribosomal protein genes, right, those are also like highly conserved. Those can also be used as additional evidence to give the correct taxonomic as well as phylogenetic position to the bacteria that you are dealing with. Okay, so typically this 16S ribosomal RNA gene is 1.5 kb gene, and generally, like when you are going for a complete genome, you should try to amplify have the primers from the almost start and end, like 27F and 1492R, right, and basically that will cover the entire 16S ribosomal RNA gene, and which can be easily sequenced by the Sanger sequencing, like 800 base pair from both sides, forward and reverse. So while you are doing the Sanger sequencing, you have to give the DNA as well as the primers, right, the forward and reverse primer. So basically you can concatenate based on the overlap that you find between the forward and reverse read, and get the complete 16S ribosomal RNA gene, and then take this gene. So there is a database in NCBI, okay, site. There is a database of all the 16S ribosomal RNA which are known from various bacteria. For your 16S ribosomal RNA, you will be doing a blast against the NCBI 16S ribosomal RNA database, which will be telling you that to which particular known 16S ribosomal RNA your ribosomal RNA is close to, okay. So if it is very close, like 97% and more, so you should know that that bacteria that you are getting the hit in the NCBI almost 97% could be the bacteria that is there in your hand, right, and if that the bacterial database is sequenced or not, you will also know from there, because if it is already there and also isolated from the same environment and there is not much difference between the two, then there is no point basically sequencing is again, okay. Otherwise, like if you find it too novel, like 90% of the identity and lesser coverage, then you should aim for the sequencing, and if that bacteria is relevant to in terms of its isolation or like the region from which you have got that bacteria, right. So basically in this lecture we looked at what are the prokaryotic genomes, the bacterial genomes, what are the common sequencing technologies, what is the best practices in genome sequencing technologies for the bacteria, the assembly approaches to construct the draft and complete genomes, and also what are the basics of the taxonomic identification, okay. So we end this lecture here today, and thank you.